So in this slide, I will explain the chapter 6. The title is the Mono-allelic expressions. Chapter 6 is composed of two subjects. First one is the genomic imprinting, and second one is X inactivation. I will explain the difference between biallelic expression and the monoallelic expression. So what is biallelic expression? Biallelic expression means that both alleles can be expressed. Okay, so what is allele? Okay, for example, so this is the female gamete and this is the male gamete. So after fertilization, they will make the diploid. Okay, the diploid offspring contains a homologous pairs of chromosomes. Okay, for example, our human has the 22 pairs of autosome, and plus women has XX chromosome and the male has XY chromosome. Okay, one set, or for example, this is chromosome 1 and this is chromosome 2. Okay, so one set of the chromosome comes from mother, and one set of chromosome comes from father. So allele is that one of two forms of a given gene. For example, some specific gene is located in chromosome 2, especially in the upper arm. Okay, so allele large A comes from the mother. Okay, they have the large A allele. At the same location, for example, small a allele, small a comes from father. Okay, so in this case, we call it as a large A, small a, or in this location, large A also can come to here. So their genotype is large A, large A. So in these cases, their genotype is large A, small a. Okay, so biallelic expression means that both large A or small a, both alleles can be expressed. Okay, however, the monoallelic expression means that, so this is monoallelic expression, only one allele can be expressed. So only large A or only small a can be expressed. Okay, so the expression is dependent on parental origin. This is, we call it as imprinting. Or, so randomly chosen allele. For example, the very famous example is X inactivation. So in this chapter, we will cover the imprinting and the X inactivation. Okay? In this slide, I will explain the first subject, genomic imprinting. So what is genomic imprinting? So genomic imprinting is a selective expression of alleles inherited from father or mother. Okay? And father and mother, they mate each other. After that, they will make their daughter or son. But in these cases, the allele inherited from father is selectively expressed in daughter and son. How about in this case? So allele inherited from the mother is selectively expressed in daughter and son. So in these cases, we call it as genomic imprinting. So parthenogenesis, in Korean we call it as the 전여생식, is an example of the genomic imprinting. So parthenogenesis is the reproduction without fertilization. For example, in sexual reproductions, so after meiosis, female make the egg, haploid egg, and the male make the haploid sperm. So after that, they will make after fertilization, they will make the diploid zygote. After this diploid zygote is developed into the diploid individual. So this is, we call it as sexual reproductions. So reproduction happens without fertilization. However, the haploid parthenogenesis, so after meiosis, so oocyte is developed. Okay, so they will make the haploid zygote. Okay, so without fertilization, so reproduction happens without fertilization. So this haploid zygote is developed into haploid individual. So this is, we call it as the parthenogenesis. In Korean, we call it as 처녀생식. So in these cases, selective expression of alleles inherited from father or mother is happen. This parthenogenesis occurs in the bees. For example, female bees make the egg. After meiosis, they will make the egg. Okay. So this egg can be developed into the haploid zygote. So without fertilization, so this egg will become the male bee. So in these cases, selective expression of allele inherited from mother happens. So this is, we call it as haploid parthenogenesis. Okay, so parthenogenesis is also an example of genomic imprinting. Only selective expression of allele inherited from the father or mother happens. Okay. So in this slide, I will explain the IGF2 gene as an example of genomic imprinting. So what is IGF2 gene? The full name is insulin-like growth factor 2. So this gene plays an important role in the growth and development of fetus. Okay, so this is the female mouse, and this mouse is pregnant. Okay, she has a fetus, and the fetus is connected in the placenta. Placenta, we call it as, in Korean, we call it as 태반. And actually the IGF2, main function of IGF2 is increase the placenta weight, and also it can increase the glucose transport and the amino acid transport in the placenta. So finally, this IGF2 gene plays an important role in the growth and the development of fetus. Okay. However, IGF2 gene has some characteristics. Okay. For example, the paternal allele of IGF2 can be actively expressed in the fetus. However, maternal allele of IGF2 is completely silent in the fetus. So we will see some detailed mechanism in the next slide. So in this slide, I will explain the genomic imprinting mechanism in the IGF2 gene. Okay, let me start maternal allele of IGF2. In next slide, I will explain the paternal allele of the IGF2. Okay, so this is the genomic region of the IGF2. So next to the IGF2 gene, there is the H19 gene is located next to the IGF2 gene. So between the IGF2 and the H19, there is a ICR. ICR is the imprinting control region. Okay, so imprinting control region contains methylation sensitive CTCF binding site. So in chapter 5, we learned that what is CTCF. So CTCF is chromatin insulator. Okay. So if in the maternal allele of IGF2, so there is no maturation in the ICR, at that CTCF can strongly bind to the ICR. So this binding is activate H19 gene, H19 activation. Okay, so H19 gene is activated. Okay, however, this CTCF binding in the ICR region is blocks, so blocks the interaction between the enhancer and the IGF2 promoter. Okay, in chapter 5 we learned that CTCF, one of CTCF function is blocks the interaction between the enhancer and promoter. So in this case, CTCF blocks the interaction between the enhancer and the promoter. So usually it inhibits transcription. So in this case, the maternal allele of IGF2, CTCF blocks the interaction between the enhancer and IGF2 promoter. So that's why the maternal allele of IGF2 becomes completely silent in the fetus. Okay. In paternal allele of IGF2, especially the ICR region, the DNA is methylated. So DNA methylation in the ICR, especially in paternal allele of IGF2. It means that in this case, CTCF cannot bind to the ICR. It means that there is no H19 activation. Okay, because there is no CTCF binding in the ICR region, and interaction between the enhancer and the IGF2 promoter happens. That's why the paternal allele of IGF2 becomes active in fetus. Okay. So in this slide, I will explain one example in genomic imprinting mechanism in IGF2 gene. Okay, so this mouse has the two IGF2 gene. One comes from father, the other comes from mother. However, the paternal allele of IGF2 is normal, but the maternal allele of IGF2 is mutant form. However, in this case, so only the paternal allele, which is the normal IGF2, so normal IGF2 can be expressed. So in this case, this mouse is received enough glucose and enough amino acids during the fetus. So that's why their phenotype is the normal size mouse. Okay. However, in this case, another case is that paternal allele of IGF2 is mutant, and the maternal allele of IGF2 is normal. However, in this case, only paternal allele can be expressed. Paternal allele is the IGF2 mutant form. Okay, that's why the mutant form is expressed. It means that there is no enough glucose transport and amino acid transport during fetus. That's why the mouse phenotype is very small. So we call it as a dwarf mouse. Okay. So in this slide, I will explain the AS gene as an example of genomic imprinting. So what is AS gene? The full name of AS gene is Angelman syndrome gene. So this gene plays important role in nervous system, and also this AS gene is located in chromosome 15. So chromosome 15 composed of the upper arm and the lower arm. So AS gene is located in the lower arm, okay, here. Okay, however, the paternal allele of AS gene is completely silent due to genomic imprinting. So on the contrary, the maternal allele of AS gene is actively expressed. They are active. Okay, so the Angelman syndrome, this is the phenotype of Angelman syndrome is intellectual disability, and they have the seizure, and also there are several phenotypes. So Angelman syndrome is caused by the maternal allele of AS gene is deleted or mutated. So maternal allele of AS gene is when deleted or mutated, so they show that the Angelman syndrome, okay, because the paternal allele of AS gene is completely silent. Okay, so this is the I show the example, the AS gene as an example of genomic imprinting. So in this slide, I will explain the PW gene as an example of genomic imprinting. So what is PW gene? The full name of PW gene is Prader-Willi syndrome gene. So this gene plays an important role in normal development. And also this gene is located in chromosome 15. Okay, so the PW gene is located here, and AS gene is located here. Okay, and the paternal allele of PW gene can be actively expressed. However, maternal allele of PW gene is completely silent due to the genomic imprinting. Okay, the Prader-Willi syndrome is caused by the paternal allele of PW gene is deleted or mutated. So when the paternal allele of PW gene is deleted or mutated, at that case, Prader-Willi syndrome happens. So this is the symptom of Prader-Willi syndrome. If they have this syndrome, they show the obesity, and also they have small hands and small feet, and their stature is very short, and they also have the mental retardation. Okay. So in this slide, I will explain the one case in the Angelman syndrome. Okay. The case is that sperm, this is sperm. Sperm has the normal chromosome 15. So they have the PW gene, they have the AS gene. However, the egg has chromosome 15, in which PW and AS genes are deleted. Okay, after fertilization, so this is their chromosome. This comes from father, paternal, and this comes from mother, it's maternal. So after fertilization, the paternal allele of AS gene is completely silent due to the genomic imprinting. So AS gene cannot be expressed. So in this case, they will induce Angelman syndrome. However, even though PW gene is deleted, however, paternal allele of PW gene can be actively expressed, and in this case cannot induce Prader-Willi syndrome, even though PW gene is deleted. Okay. So in this slide, I will show you another case in Prader-Willi syndrome. Okay. In this case, sperm has chromosome 15. sperm has chromosome 15 in which PW gene and AS genes are deleted. However, egg has the normal chromosome 15, so they have the PW gene and they have the AS gene. So apt fertilization, so this is apt fertilization, okay, apt fertilization, okay, maternal allele of PW gene, so maternal allele of PW gene is completely silent due to the genomic imprinting. So in this case, so they will induce Prader-Willi syndrome. However, in this case, the AS gene is deleted. Even though AS gene is deleted in the paternal allele, however, maternal alleles of the AS gene can be actively expressed. So in this case, they will not induce Angelman syndrome. In this case, they will only induce Prader-Willi syndrome. Okay? So in this slide, I will explain the second subject, X inactivation. The full name of X inactivation is X chromosome inactivation, XCI. So X chromosome inactivation means that random inactivation of one of XX in the females. Previously, we learned that this concept in chapter 4, the dosage compensation in the murmur. So dosage compensation in mammalian happen in female XX. At that time, we also learned that random inactivation of one of XX chromosome happens in the mammalian. But at that time, we didn't cover the detailed mechanism. But in this chapter, we will cover some detailed mechanism. Okay, so in this slide, I will briefly cover the XCI mechanism. Okay, so cyst, this non-coding RNA, cyst non-coding RNA initiate heterochromatin formation, and also this cyst non-coding RNA induces heterochromatin spreading to whole X chromosome. So this is the XCI mechanism. But we will see some details mechanism from next slide. In this slide, I will explain the X inactivation center, XIC. So this is the X chromosome. So X chromosome has the XIC. Okay. So XIC means that is where cyst non-coding RNA is exist. This is a cyst non-coding RNA. Okay, the size is about 680 kb to 1.2 megabase in size, and XIC is consists of the 5 non-coding RNAs. Okay, first one is JPX, and the other one is cyst, and the other one is also TSIX. And also there is the XITE and also TSX non-coding RNAs. So mainly two non-coding RNA, cyst and TSIX, are mainly expressed in this XIC. These two non-coding RNAs are mainly expressed. But especially depending on the presence or the absence of the cyst non-coding RNA, so X chromosome becomes XI, inactive X chromosome, or X chromosome becomes XA, so active X chromosome. Okay, so table 6.1 shows the summary of XIC element. Okay, And the main element of XIC is the cyst and the TSIX. Okay, so the cyst is the non-coding RNA, is located here, and the TSIX is also non-coding RNA. Okay, so main function of cyst is its main function is the random inactivation of one of X chromosome, and also TSIX is their main function is that this is the antisense RNA of cyst non-coding RNA. So TSIX is the antisense RNA of the cyst non-coding RNA, and the main function is the TSIX induces transcriptional inactivation of cyst. We will cover this concept in the next slide. Okay, so you also have to see the several components here. So, you don't have to memorize the function. Okay. So in this slide, I will explain the three models of X chromosome inactivation. Okay, so the first model is sensing and choice model. So this model is the most trusted model. Okay, and also this model used cyst, the non-coding RNA. Okay, so during the early embryonic state, cells start to differentiate. At that time, two X pairing region, XPR, two XPR is pairs with each other. So two XPRs pairs, that this pairing upregulate the cyst on both X chromosome. Okay, at that there is a two TSIX and XIT region. So this region is the cyst region. So next to the cyst region, there is a TSIX and also XITE region. So this region is pairs each other. Two TSIX and XITE region is pairs each other. Okay. So after that, choose which X chromosome to activate or choose which X chromosome to inactivate. So in this case, they choose this X chromosome to activate and choose this X chromosome to inactivate. Okay, so after that, in this X chromosome, TSIX repressed cyst expression on X chromosome. After that, this X chromosome remains active. However, in this X chromosome, TSIX does not repress the cyst expression on the X chromosome. That's why in this X chromosomes remains inactive. So this is the sensing and choice model, and this model is most trusted model in the X chromosome inactivation. So in this slide, I will explain the second model of X chromosome inactivation. The second model is blocking factor models. There is no involvement of cyst non-coding RNAs. So our human chromosome have the autosome and the sex chromosome. And also, for example, in this case, this autosome creates a blocking vector, single blocking factor. So autosomes work as a diploid pair to create a single blocking factor. So they made the single blocking factor. So after that, this blocking factor is bound to the X chromosome, one of X chromosome. Okay, so X chromosome bound by the blocking factors remains active. However, the remaining unbounded X chromosome, the remaining X chromosome is inactivated. Okay, so this is recorded as the blocking factor model. There is no involvement of cis non-coding RNA. Okay, so usually the autosome, we have the 22 autosome. Okay, so autosome creates a single blocking factor, so that this blocking factor is bind to one of X chromosome. So X chromosome bound by the blocking factor remains active. However, the remaining X chromosome is inactivated. So this is the second model of X chromosome inactivation. There is no involvement of cis non-coding RNA. Okay, the third model of X chromosome inactivation, we call it as two-factor model. And this model is also, there is no involvement of cis non-coding RNAs. Okay, let me start the two-factor model. Okay, so autosome work as the diploid pair to create a single blocking factor. So autosome creates a single blocking factor, and also X chromosome another factor. So autosome creates one factor, and also X chromosome creates another factor. This is a two-factor model. So X chromosome creates two competence factors. Okay, so X chromosome creates competence factor. Okay, so blocking factor, these blocking factors neutralize the one of two competence factor. Okay, so this one, so okay, the blocking factor with competence factor is bound to the one of the X chromosome. So X chromosome with blocking factor with competence factors remains active. However, the other X chromosome, so X chromosome bound by only competence factors is inactivated. So this is also third model of X chromosome inactivation, two factor. So autosome make the one factor and X chromosome make another factor. Also, these blocking factors neutralize the one of the two competence factor, and also X chromosome with the blocking factor with competence factors remains active. However, the X chromosome bound by competence factor is inactivated. So this is the third model. Okay. On this slide, I will explain the details mechanism of X chromosome inactivation, especially focused on sensing and choice model. So in sensing and choice model, sensing is happen at that choice step is happen here. So usually the choose which X chromosome is inactive or choose which X chromosome to be inactivated. Okay, so this initiation step occurs in the X chromosome that was chosen to be inactivated. Okay, so this is the cis non-coding RNA structure, which has a lipid A motif and lipid 2B, lipid C, and lipid E motif. So especially PRC2 bind to the lipid A motif of cyst. Especially PRC2, we learned that PRC2 have the RNA binding domain. Okay, especially the RBAP48 in PRC2 bind to the lipid A motif of cyst. And also PRC2 has the EZH2 domain. So this domain is bound to the repeat A motif, and this domain induces the H3K27 methylation. So H3K27 methylation. Okay, so this is the initiation step of the X chromosome inactivation. In this slide, I will explain the transcriptional regulation of cis non-coding RNA in active X chromosome. Okay. In previous slide, in this slide, I summarized the XIC element, especially focused on the cis non-coding RNA and TSIX. And then in this slide, I will also explain another function, another element, DXPAS34. So this is DXPAS34. The main function of DXPAS34 is this is a regulating factor of TSIX. So this is the regulating factor of TSIX. Okay, so usually the DXPAS34 activates TSIX. After this TSIX, the non-coding RNA induces the transcriptional inactivation of cis non-coding RNAs. So this step happens in active X chromosome. Okay. In this slide, I will explain the transcriptional regulation of cis non-coding RNA in inactive X chromosome. So through sensing and choice model, so X chromosome was chosen to be inactivated. After that, the H3K27 methylation happened. After that, so there is no DXPAS34 activation. So it means that there is no TSIX expression. So in this case, transcription activation happens. Transcriptional activation of cis non-coding RNAs happens. Okay, after that, cis non-coding RNA is expressed. After that, just the cis non-coding RNA coats one of the XX chromosome that was chosen. Okay, cis non-coding RNA coats one of the XX chromosome and makes it as the inactive X chromosome, XI. So this is the transcriptional regulation of cis non-coding RNA in inactive X chromosome, because there was no DXPAS34 activation. It means that there is no TSIX expression. In this case, transcriptional activation of cis non-coding RNAs happens. Okay. In this slide, I will explain the maintenance of X chromosome inactivation. Okay, so this is the inactive X chromosome. For the maintenance of X chromosome inactivations, so DNMT is the DNA methyltransferase is further induced methylation. And also, usually in the nucleosome, usually the H2A is incorporated into the normal nucleosome. But in the inactive X chromosome, so macro H2A is they are nearly similar to the H2A, but size is bigger than the H2A, so that's why we call it as macro H2A. So macro H2A is incorporated in the nucleosome of the inactive X chromosome. So in this case, you will see that macro H2A accumulate at the inactive X chromosome. So usually the incorporated macro H2A interferes the binding of transcription factor and also interferes the binding of SWI/SNF. SWI/SNF is the nucleosome remodeling factor. So through this mechanism, so X chromosome inactivation can be maintained. So in this slide, I will explain the spreading of X chromosome inactivation. So we call it this mechanism as the proximity transfer, 근접 전송. Okay? Especially the cis non-coding RNA is expressed in inactive X chromosome. Okay, after that, this cis non-coding RNA forms chromatin loop. So this is chromatin, make the chromatin loop is like this, and also this induces the special proximity. So this region and this region is far away from each other, but after that the chromatin loop is made, and this loop is very close to each other. So finally, so X cis induces special proximity, 공간적 근접성 in inactive X chromosome. Okay, so after that, PRC2, especially the RBAP48 in PRC2, bind to the repeat A motif of cis, and also PRC2, especially the EZH2 proteins, induces the H3K27 methylation in nearby region of inactive X chromosome. So it means that it can spread X chromosome inactivation in the nearby region. So this is the spreading of X chromosome inactivation mechanism. So once again, we call it as the proximity transfer. So initially the cis forms chromatin loop. It will induce special proximity in inactive X chromosome. So after that, PRC2 bind to the repeat A motif of cis, and especially the EZH2 induces H3K27 methylation in nearby region of X chromosome. So spontaneously, X chromosome inactivation is spreading, it happens in nearby region of inactive X chromosome. So in this slide, I will explain a new concept, X chromosome reactivation. Okay, so usually the pluripotent stem cell have two active X chromosome, so they have XA and XA. However, when differentiation is initiated, one of X chromosome undergoes X chromosome inactivation. So that's why they will make the XI inactive X chromosome. So this is we call it as the differentiated cells. Okay, so differentiated cell is also can be changed to the pluripotent stem cell. So this procedure, we call it as reprogramming. So when reprogramming happens, differentiated cell is become to the pluripotent stem cell. So during this step, X chromosome reactivation happens. So XI will become to XA. Okay, so in next slide we will see some details mechanism. So in this slide I will explain the X chromosome reactivation during the iPSC reprogramming. Okay, so this is the fibroblast cells which has the XA and XI. Okay, so we can introduce the POL transcription factor, which is we call it as OCT4 and SOX2 and KRF4 and CMYC. So after we introduce the POL transcription factor gene in the fibroblast, the XCR happens, especially in the XI inactive X chromosome. TSIX expression happens. Okay, this TSIX induces transcriptional inactivation of cis non-coding RNA. So finally, it will make the X chromosome reactivation. So after X chromosome reactivation happens, so fibroblast will change it to the iPSC cells, induced pluripotent stem cell. So their X chromosome status is they have the active X chromosome. They also have another active X chromosome. So this is the XCR. We call it as the X chromosome reactivation. This is done by the introduction of the OCT4 and SOX2 and KRF4 and CMYC. So after that, the reprogramming happens. So during the reprogramming, XCR happens, especially the TSIX expression happens. After that, TSIX induces transcriptional inactivation of cis non-coding RNA. Finally, X chromosome reactivation happens. Okay. Today we learned the chapter 6 mono-allelic expression, especially focused on the genomic imprinting and the X inactivation. Okay. Today we will finish the class.